So when we as clinicians are now deciding on the use of immuno-oncology drugs, there are several things we look at. First and foremost, we need to know that the drug is approved for the indication. We also have to have a sense that the patient's pace of their disease is not going that rapidly that we might want to use something else first. And what I mean by that is that an immuno-oncology drug works by allowing a T cell, which is one of the immune functioning cells, to reestablish the ability to kill tumor cells. But T cells take time to work. It's not like chemotherapy where you give it and within minutes to hours the tumor is regressing. It could take, with ipilimumab, it could take up to a month to 6 weeks even to begin to work. And the anti-PD-1 drugs, Nivo and Pem, they work a little bit faster, but they don't work immediately. So you want to know that the drug is approved for the proper indication, and you also need to sense that there's time for the drugs to work. Otherwise, you may have to give another agent first or with those drugs. The use of immuno-oncology drugs in general is a new technology. It's a new field. And doctors are somewhat struggling with understanding, how do I know this drug, which is expensive and does take time to work, is going to work for my patient? So we now are starting to look for biomarkers, signs, indications that these drugs will work. Now, we know that the greater the number of mutations a cancer has, the more likely an immuno-oncology drug is going to work. There's recent data suggesting that NRAS mutations may confer greater sensitivity to the anti-PD-1 or anti-PD-L1s. We know, for example, that melanoma, which has some of the greatest number of mutations, which are genomic abnormalities, these are the DNA of the tumor cells are making aberrant proteins called neoproteins or oncoantigens that the immune system then can recognize. So the more you make, the more likely it is that the tumor will be recognized when you give one of these drugs. There's another whole class of biomarker which really hasn't panned out quite as we expected, and that is for the anti-PD-1s. You would imagine that you would want to see PD-1 expression, but it turns out that it's important in some circumstances. It's not absolutely predictive. There are patients who don't have high levels of PD-L1 or PD-1 expression in their tumor, and yet they respond to anti-PD-1s. And that may be because of sampling error or other issues. So we're at the very beginning of finding the biomarkers, but for sure, only about half of patients respond to immuno-oncology drugs when given in combinations or sequentially, and less than that when they're given alone respond. So there's a reason for that, and we don't want to be wasting the patient's time or spending money and precious resources when there's no chance for a drug to work. So biomarkers are and will become increasingly important. So one of the things that people are talking about now is the variability between diagnostic tests for biomarkers, PD-1 expression as an example. There is variability. I think, though, that even with the variability, the marker itself may not be correct. The immunologic milieu and the tumor milieu, the microenvironment of the tumor, is not a static situation. It modulates itself. And so you may be taking the wrong piece of tissue from the wrong site. It may be that the patient's tumor is leaving an inflammatory state where there would be a lot of expression of PD-1 and going to a non-inflammatory state. And we're now starting to think about how to get it back to an inflammatory state, because that's when immuno-oncology drugs work better. So there's still a level of complexity here, peeling back the onion, so to speak, that we need to do before we can answer the question of, is this biomarker better than that biomarker? Is there greater specificity with this drug, with this biomarker versus another? The field and we are not ready to answer that question yet.